Yun yung ipe-plate natin. And then we will check after 24 to 48 hours of incubation kung meron nga ba talagang tumubo na E. coli or any other type of coliform dun sa ating plate. So he's just trying to do another plate kasi dalawa yung kanyang culture meter. So that's for filtration. So next, we'll continue with MPN. So for that of the most probable number method, it would be statistical estimation lang ng ating microorganisms that would be grown dun sa ating culture medium. So for that of MPN, this would be discussed further in water bacteriology. So later, it would actually be discussed, lalo na kapag pupunta na tayo dun sa ating mga coliform microorganisms and the other Enterobacteriaceae. So it's based on the fact that the greater the number of bacteria in a sample, the more dilution is needed to reduce the density to the point at which no bacteria will be left to grow in tubes in a dilution series. So similar, somehow similar to that of yung ating serial dilution agar plate natin na technique. And for direct microscopic count, it's measured volume of a bacterial suspension that will be placed within a defined area on a microscope slide. But this is not usually done in microbiology. But in certain kasi ng mga antawag dito, facilities, especially those that would be for production ng milk, ginagawa nila to. But since clinical tayo, hindi natin to ginagawa. But this particular device, your counting chamber, it is similar to that of your hemocytometer, which you will be utilizing in your hematology for counting that of your CBC, your particularly your RBC at saka your white blood cell. So we would actually be utilizing a cell counter. So yung mga cells, yung mga bacterial cells, if it would actually be in the case of microbiology, those particular cells that would actually be present in one particular grid or large square, yun po yung bibilangin natin. Wait lang, I'll check kung meron akong video for that. Oh yeah, meron. Tapos na ba kayo with, asa na pala kayo sa HEMA? Baka kasi mamaya na over po itong discussion na to. Ano na bang topic ninyo sa HEMA? Sorry. Okay. Matagal pa nga. But, just to show you lang how to do ang tawag dito, a bacterial counting with the use of your counting chamber, which is somehow similar to that of your hemocytometer. So we tend to mix that of the inoculum with that of a dye para mas mabilis or mas madali yung ating trabaho. Kasi again, yung katulad ng sinasabi niya, it would actually be utilized for distinguishing your living at saka your non-living cells. Kasi di ba, mas reliable naman talaga kapag ang in-include lang natin yung viable, yung living natin na cells. Kasi as healthcare workers, yun talaga yung pinaka-tina-target natin in order to prevent that of the chain of infection and the spread of disease or infection. So yun, you're trying to load, ang procedure kasi yung tawag dyan would actually be loading that of the hemocytometer. Mind you guys, it's not that easy talaga to do this. Kasi, kailangan ganito siya. Nakikita nyo paano nyo ginawa. It's very smooth lang dapat yung paggawa mo. Paglagay mo ng sample dito sa chamber na to. And, i-avoid nyo ang bubbles, i-avoid nyo din ang isang dead space. Dapat buong chamber na yan ay napuno. And, don't spill yung ating inoculum dito sa may moat. This is what we call as the moat. Itong mga nasa side niya, yung mga hollow areas dito sa side. Pwede kasi mahulog dyan yung ating sample. At kapag nangyari yon, again, repeat mo ulit yung loading process. Dapat walang mahuhulog, may spill dun sa ating moat. Alam niyo naman yung moat, di ba? Sa Middle Ages, yung parang tubig around a particular castle, that is what we call a moat. And ganun din yung nandito. So again, avoid bubbles, avoid an empty space, and avoid spilling it into that of the moat. And dapat smooth yung pag-dispense ninyo, loading ninyo sa ating hemocytometer. Ganyan din po yung gagawin for that of hematology. So nakikita nyo, ang ganda ng pag-load niya. At kailangan, steady lang yung kamay mo dyan. Bawal yung nanginginig. So once it would actually be loaded, you wait muna for minutes. Usually enough na ang 1 to 2 minutes in order for the cells that are present dun sa ating mixture to settle dun sa ating chamber. So wag agad-agad na titignan dun sa ating microscope. Wait mo muna na mag-settle sila, mag-sediment dun sa ating chamber. So, when trying to count cells in hemocytometer, ganito din po yung sa hema. You only will include the cells in the inverted L natin na area. So, this is your inverted L. And yung mga cells that are included in this particular area, they are counted. Pero yung mga nag-touch dito sa mga grid lines, on that of the other side, hindi na po yan bibilangin. Ang tanong minsan, paano kapag dito sa ating inverted L, kunyari dito sa ating inverted L ha, yung cell natin, kasi dito sa inverted L, meron din siyang mga grid lines. We would actually have ito, parang 3 grid lines. Would you still count yung mga nandito sa third portion or outer? Kapag lumabas na siya dun sa grid line, always remember, if lumabas na siya dun sa outermost grid line, kahit nasa L portion na siya, usually hindi na natin ini-include. Kasi labas na po siya dun sa mismong quadrant, dun sa mismong large square. But that will be discussed better in that of your hematology. So again, dapat matalas yung mata. Try to remove or don't include in the counting yung mga artifacts. So ganun lang, bilangin mo siya in that of the large square. And then get the total after. So that is actually how to use your hemocytometer and also in the calculation of the viable cells. Any questions? Any questions? Okay. So for estimating naman bacterial numbers through indirect methods, we have your turbidity, but this is actually done usually for liquid natin na media or yung mga, ating mga broth. So for some types of experimental work, estimating turbidity is a practical way of monitoring that of your bacterial growth. And as bacteria multiply in a liquid medium, it then becomes hazy or turbid or cloudy with certain cellular elements that are actually suspended or moving dun sa ating liquid medium. Tapos na ba kayo sa introduction to urinalysis sa AUVF? Guys?
Yes, ma'am.
Yes.
Okay, di ba sabi doon, ang tawag na dito, di ba sabi doon na the changes in a 24-hour unpreserved urine specimen would actually be indicated by turbidity din as well. Kasi meron tayong bacterial multiplication, di ba? So kapag mas marami pong growth, mas maraming bacteria ang nagawa or nabuo, that would then increase yung ating cellular density. Sobrang daming bacterial cells that are suspended in that of the liquid medium or in that of the broth. Thus, mas magiging turbid siya, mas magiging opaque, mas magiging cloudy, mas magiging hazy yung itsura niya. So the instrument that is being utilized to measure your turbidity, that is your spectral photometer. I think this would be discussed better in that of your clinical chemistry. Tapos na ba kayo with instrumentation? Or papunta pa lang kayo doon?
Papunta pa lang po.
Okay. So for that of your spectral photometer, this beam of light that is actually being emitted by that of the light source, it will then be deflected by the bacterial cells that are suspended dun sa ating tube medium, dun sa ating liquid broth. So, yung na-deflect na light, yun yung ultimately babasahin ng ating reader, ng ating spectral photometer reader. So, di ba na-deflect siya? And those that would actually be able to pass through, yun lang po yung madedetect ng ating spectral photometer reader rather, and it would actually be presented in the detector. So an example of your liquid medium na usually nilalagay natin for that of the spectrum for testing ng ating turbidity. Again, we have your Macfarlane standard na ginagamit natin for antimicrobial susceptibility testing. And we also have your nephelometric method, but this is actually more for clinical chemistry. Next, we have your metabolic activity. It assumes that the amount of certain metabolic products like acids or carbon dioxides, they would actually be directly proportional to the number of the bacteria that would be present. So for metabolic activity, this would be discussed better once you would go sa biochemical test. Kasi yung amount ng acid na nagawa, yung amount ng carbon dioxide din na nagawa, and any other type of metabolites, they can actually be correlated with that of the number of bacteria that are present in one particular colony. Depending kasi on the intensity nung kulay, nung changes ng mga color associated with acids, with carbon dioxide, with sugar, so yung mga yon. An example of a practical application of metabolic test is the microbiological assay in which acid production is used to determine the amount of vitamins. But that's not the case for clinical micro. Mas kailangan natin siya for that of identification ng ating microorganism. Next, we have your dry weight. This is more associated with your mycology, hindi masyado sa bacte. So one of the better ways to measure the growth of filamentous organisms would be via dry weight. So in this procedure, the fungus is removed from the growth medium. It will then be filtered in order to remove yung mga extraneous material and then dried in a desiccator and then eventually, iwawain natin siya. Ganun din for that of bacteria. But again, hindi siya madalas ginagamit or ginagawa for bacteriology. Madalas siya ginagawa for your mycology. So that's it for the first part of yung ating lesson for today. Next, we proceed with isolation and cultivation. 15 minutes break. Let's resume at 10 o'clock. Okay ba yun sa inyo?
Yes, ma'am.
Okay.
Yes, ma'am.
Sigurado kayo, wala na kayong tanong ha? Dun sa
Yes, ma'am.
Okay. Yung hemocytometer, okay tayo doon. But, again, hindi siya madalas ginagawa sa micro. It would be more for your hematology. Okay? So, but at least alam niyo kung paano siya. Nakita niyo naman sa video, di ba kanina, kung paano siya i-load, paano siya, yung binibilang, yung inverted L. So, more likely, yun lang naman yung pagkakapareho niya rather, dun sa ating hematology na hemocytometer. So, let's continue with cultivation. So, do you have a copy of bacterial cultivation? Wala pa.
Wala pa po, ma'am.
Sige. Just please do make the necessary corrections or necessary na addition dun sa module. Sila yung mga naka-highlight ng red. So, why do we always do bacterial cultivation? Ito yung madalas na ginagawa natin sa ating microlab. So, it would be for growing and isolating all bacteria that would be present in a clinical specimen. Alam naman natin na ang isang clinical specimen coming from the words, hindi naman talaga siya malinis in the sense na iisa lang yung microorganism na nandoon. It will have multiple microorganisms and a lot of contaminants, especially if hindi talaga siya na-collect ng mabuti. Also, for bacterial cultivation, it would be a way of determining which of the bacteria that grow are most likely causing infection at kung sino yung mga contaminants lang or colonizers or part ng ating normal flora na hindi naman significant, hindi katulad ng mga pathogenic na microorganisms. It would also be to obtain sufficient growth of clinically relevant bacteria, again to allow identification, characterization, and susceptibility testing. So we actually have different types of culture and we have different terms that we need to understand. So we have your pure culture. Again, kapag pure culture, it will just contain a single species of microorganism. According to Bailey and Scott, a pure culture would also be an organism coming from the same genus, same species. It would have similar or identical genetic and phenotypic characteristics. Alam niyo naman, di ba, ang pagkakaiba ng phenotype at saka ng ating genotype? Tama?
Yes, ma'am.
Okay.
Opo.
Asaan si Dalapol? Joward. Joward, nandiyan ka ba? Joward, nandiyan ka pa ba? Hello?
Research. Research. 62.
Andiyan pa ba si Joward? Joward? Anyway, hanap na lang tayo ng iba. Conrada, Marlo, andiyan ka ba?
Yes po, ma'am.
Okay, what is the difference ulit ng genotype and phenotype? Basic genetics?
Ma'am, uli po, ma'am.
Anong difference ng phenotype sa genotype?
Ma'am, yung ano po, yung phenotype po is yung sa may physical characteristics po ng ano, tapos sa may
Yung genotype?
po is yung sa may ano po, sa particular gene po ata yun. Yung genetic characteristic po sa organism po yung genotype.
Okay. So, for that of phenotype, tama yun, that is your physical makeup. Kung anong nakikita mo sa isang individual, sa isang organism, yun na yun. And then, for the genetic makeup, yun yung ating genotype. Ano yung genetic makeup? Kung ano yung nandoon sa kanyang nucleic acid, sa kanyang DNA material. That is your genetic makeup. And it would all make the difference, again, yung sa genetic makeup, because of DNA exchange. Ano na ulit yung tatlo na mechanism to do DNA exchange ng ating microorganism? Ano yung tatlong means of doing DNA exchange ng ating bacteria? Guys. Ano na yung tatlo?
Transformation.
Okay. Saka?
Conjugation.
Okay. Diyan sa tatlong yan, sino yung pinaka-associated with antimicrobial resistance?
Transformation, ma'am.
Your
Transformation.
conjugation. Siya po yung pinaka-associated with antibiotic resistance, yung ating conjugation. At sa conjugation, ano na ulit yung genetic material that is needed to produce your sex pili and the other enzymes and proteins for that of the process of conjugation. Ano na yung genetic material na yun?
Plasmid, ma'am.
Okay, your F plasmid. Very good. Okay. Sino naman yung vector natin for that of transduction? Yung nagtatransfer ng genetic material?
Bacterial phage, ma'am.
Okay, very good. Ready na for exam next week. So next, again, always remember, pag pure culture, isa lang po siya na species coming from the same genus, same species, pare-pareho yung kanyang genetic and phenotypic characteristic. Wag na wag kakalimutan yan. So the process of cultivation, that would be, wait lang. So for the process of cultivation, that would be the process of growing microorganisms in culture by taking bacteria from the infection site, which is regarded as your in vivo environment, kasi nandoon siya sa kanyang natural habitat, dun sa mismong sugat, dun sa mismong abscess, dun sa mismong nana. So that is actually your in vivo environment. So taking bacteria from the infection site by some means of specimen collection procedures and growing them in an artificial environment in the laboratory. Ito yung ating culture media or dun sa ating mga tube medium. That's actually being regarded as your in vitro environment, which is a controlled environment for the growth of a microorganism inside the clinical laboratory. So increasing the population of microorganisms by providing their nutritional and physical requirements. So nutrients, like any other organism in the globe, it would actually need certain nutrients for it to grow. So yan yung napag-aralan natin dun sa last module. So extracellular substances that will then provide the cell with materials for building that of their protoplasm, their structure, their bacterial structure, and for creating that of their ATP, for energy generation. So a culture medium is any material for growth and cultivation of microorganisms, and it will then be enriched with that of carbon, nitrogen, sulfur, phosphorus, hydrogen, oxygen, and a buffer. So meron siyang organic, inorganic substances, and meron din siyang buffer in order to counter or minimize that of the effect of excessive pH changes. Ano na ulit yung tamang pH natin for growing that of a microorganism? Anong pH na yung kinakailangan natin?
Between 6.5 and 7.5 po, ma'am.
Okay, it's always in the range near neutrality, near that of the pH of 7. That is actually for bacteria. Magkakaiba po siya for different microorganisms, like your fungi and your viruses. Okay. And it would also be equipped with inhibitory agents. Bakit ba kinakailangan natin ulit ng inhibitory agents? That is actually in order to promote the growth of a desired, yung tina-target nating organism, at isuppress, ipahina, or prevent the growth of the other organisms, lalong-lalo na yung ating mga contaminants or colonizers or yung ating normal flora. Kung gusto lang kasi nating ipatubo yung ating pathogenic microorganism. Kaya nagdadagdag tayo ng mga agents, ng mga nutrients, ng mga substances that will inhibit the growth of the other organisms. And then we have your inoculum. These are microorganisms that are introduced into a culture medium to initiate that of the growth of that particular organism. And culture, microbes that grow and multiply in dun sa loob mismo ng ating culture medium, if it's actually a broth or a liquid one, or on dun lang sa surface for that of a solid agar natin na medium. So we actually have different types of a culture. We have again your pure culture, kapag again iisa lang po yung ating species. Kapag mix containing multiple natin na microorganisms, and then stock natin na culture. So kapag stock, it would be composed of several species that are placed in different culture medium. So usually one species per culture medium, and then it will be incubated, it will then be stored in a stock-like manner. So parang pinagpapatong-patong lang siya kapag ini-store siya, kapag ini-incubate siya. So grown in a large volume of broth and then divided into small freezer vials in order to lengthen the shelf life of the specimen to at least a year. So usually, ito yung mga ginagawa for research laboratories. Nagpapatubo sila ng mga microorganisms na ultimately gagamitin nila for different researches. So we have your agar or your agarose gel, which is the most common solidifying agent. Ay matigas, medyo matigas yung ating culture medium. So it's a sulfated polymer made up of D-galactose, 3,6-anhydro-L-galactose, and D-glucuronic acid. And usually it is derived from your red algae. So it is actually it would actually have this unique characteristic of being able to melt at 80 to 90 degrees and solidify at 40 to 50. But do take note that at 55 to 60, that is the cooling temperature for the distribution of culture medium na nai-transfer na po dun sa ating Petri dish. And usually, 20 to 25% yung amount of molten agar, yung liquid natin na agar, na itatransfer dun sa ating Petri dish for solidification. So the different use of culture media, again, it's for the growth and the maintenance of microbial culture, to favor the production of particular compounds, and to study microbial action on some constituents, different components ng ating culture medium. So we have different types of your culture medium depending on different parameters. So based on the consistency, we have your liquid, semi-solid, and that of the solid. So kapag liquid, it is commonly known as your broth medium. So kapag broth medium or liquid medium, it does not contain a solidifying agent. Wala siyang agar. But the nutrients, they are suspended, they are mixed in a liquid natin na medium. So the enrichment properties of the media enable microorganisms to be recovered in higher number. And again, its detection, yung number ng microorganisms that are suspended in it, they would actually be, most of the time, detected through that of your spectral photometer. So, talagang kinakount natin yung ating number ng ating microorganisms based on the turbidity, yung haziness na magagawa nila. And that can actually be counted through that of your spectral photometer. So, it's used for detecting the presence of a low number of microorganisms that may be missed in a plated solid agar. So it has specialized formulas that may be used for biochemical testing. So it can be counted also with that of your colony forming units per ml. It would allow the growth of aerobes, anaerobes, and your facultative anaerobes. Clear naman tayo, di ba, dyan sa different types ng microorganism na yan based on their oxygen requirement? Yes or no? So, sige nga. Kapag aerobes, saan mo usually nakikita yung haziness, yung cloudiness? Sa part ng ating tube? Kapag aerobe, obligate, aerobe.
Sa may surface, ma'am.
Okay, dun siya sa may top portion ng ating tube. Paano naman kapag obligate anaerobe?
Bottom po.
Sa may? Okay, sa may bottom. Bottom na portion. Bakit? Ano na bang effect ng ating water or ng any other type of liquid dun sa oxygen? Water acts as a action ng water with regards to that of oxygen or any other gaseous element for that matter? Di ba sabi natin, water acts as a barrier, di ba? it would actually try to prevent that of oxygen to going deeper into that of the floor or the lower portion of the tube. Diba? Once na, lalo na yung mga deep divers, if you have actually noticed, aside from pressure dun sa depths of the ocean, as they would actually descend deeper and deeper into that of the ocean floor, less and less po yung ating oxygen. And that of also increase yung kanyang pressure. Kaya mahirap huminga dun sa pinakamababang part ng ocean. Ganon din po for that of the different organisms. They will not be able to get enough oxygen kapag nandoon sila sa pinakamababang part ng ating test tube. Saan mo naman makikita yung ating mga facultative aerobes? Saan part sila ng tube? Hello? Nandiyan pa ba kayo?
Ano na po ito?
So yun, paano kapag facultative aerobes, saan sila nakasuspend dun sa ating test tube?
Throughout.
Okay, it would actually then be distributed throughout that of the tube. Paano naman yung mga aerophilic, aerotolerant organisms? Saan sila makikita? Ano?
Kalat-kalat.
They would actually be distributed again all throughout the yes, that of the test tube because they can actually tolerate fluctuations ng oxygen. Paano yung microaerophilic organisms? Nasaan sila sa test tube?
Sa top na.
Sa may?
Medyo top ng tuba.
They would actually be
Sa may
located
liver.
in the middle, yes. They would actually be located in the middle because they would actually only require a small amount of your oxygen. So wag na wag kakalimutan yung mga basic information na yun na yun yung mga must know. And you would actually be reading kasi try to classify yung mga binabasa ninyo as the basic must know at saka yung mga nice to know. Kasi sa sobrang dami ng subjects ninyo, sa sobrang dami ng mga topics din na dinidiscuss each and every day, yung mga must know, yung mga basic, please lang, wag na wag ninyong kakalimutan yun up until the day of the board exam. Kasi whether it be given as a direct question and answer lang, identification, or bibigyan ka ng case, yung mga basic concepts na yun, always tinatanong sila. Kasi nga, basic sila. They are must know. So, an example of your liquid medium would be your nutrient broth, your brain heart infusion, your trypticase soy, and your thioglycolate. May encounter pa natin yan later as we progress in this topic or in this module. Next, we have your semi-solid medium. So, semi-solid siya kasi onti lang po yung solidifying agent niya. It would just contain 0.5 to 1% of your agar. So, it has a consistency between the liquid and the solid. So, para siyang jelly natin na consistency. It's not as hard as your solid natin na medium, and it's not as flowy as that of your liquid medium. It has a lower concentration of your agar than solid medium, and it's commonly used in determining the motility of an organism. Kasi it's more loose as compared to the rigid, very hard natin na solid medium. So, it would also be needed for detecting indole at saka yung sulfide production, which we will tackle in biochemical tests. So, an example would be your sulfide indole motility medium. So, an example itong picture show your SIM motility. So this is an example of a butt medium. A tube medium kasi would actually be divided into three: butt, slant, and your butt slant. So this is an example of a butt medium. It has a flattened natin na distribution or dispensing of your medium into that of a test tube. So ang gagawin, always remember, kapag butt medium yan, ang gagawin natin, we stab. Tutusukin mo siya with an inoculating needle up until 2/3 ng ating tube. Wag hanggang dun sa dulo, hanggang 2/3 lang po. So we stab, tutusukin mo siya ng ating inoculating needle. And then, incubate it overnight, usually 24 to 48 hours. And then, take note, if there would be haziness or cloudiness around the stab area, or yung buong area would actually be hazy or cloudy, that is positive. It means meron tayong motile, masyadong magalaw na organism dun sa loob. Kasi gumalaw siya away from the stab area, and it actually caused a haziness or turbidity around that of the stab area, sometimes involving that of the entire tube medium. So that would actually be positive as compared to this one. Nakita nyo, di ba? Dun lang sa may stab area, yun lang po yung nakita po natin, hindi gumalaw yung ating microorganism. So it's non-motile. Dun lang siya sa may stab area. Pero dito, hindi mo na makita yung stab area eh. Yung buong medium would actually be involved. It would actually be very turbid. So it is a highly motile organism. Na clear tayo sa ating SIM?
Yes po.
Okay, and next we have your solid one. That would actually be composed of 1.5 to 2% agar. It would actually kasi contain the solidifying agent, your agarose gel or your agar. And it does not inhibit bacterium or liquefy at room temperature. It is actually used for developing surface colony growth of microorganisms. It is critical for obtaining isolated colonies. Depending kasi on the type of solid natin na medium, yung iba, it would actually be able to promote the growth of all microorganisms. Wala siyang ini-inhibit. Pinapatubo niya lahat ng microorganism. Yung iba naman, nagdadagdag tayo ng ating inhibitory agent. It will just promote the growth of selected microorganism. Tapos ini-inhibit niya yung growth ng iba natin na organisms. So an example of your solid medium, we have your PSI, your triple sugar iron, your MAC conky, your BUP, and your cup, your blood agar, and your chocolate agar. So mamaya, ma-discuss natin yan further. So based on composition, we have naman your synthetic and the non-synthetic. Kapag synthetic or defined medium, kaya siya tinatawag na defined kasi alam natin yung kanyang component. So all components are chemically defined, exact chemical composition of ingredients would actually be known. Ito yung mga commercially available na culture media. Binebenta na siya as it is. Alam na natin kung ano yung components niya. This is actually utilized for research purposes, yung mga research facilities, not in clinical hospital na type or scenario or setting. So they would actually be present either in a liquid medium or solid medium. They are preferred for the isolation of your cyanobacteria and your chemoorganotropes, like your BG11 medium. For your non-synthetic or complex medium, ito yung more likely ginagamit natin sa hospital or sa mga clinics. So it will not contain